We're going to talk a little bit about some breathing gas pathway devices, but before we get started, I thought I would introduce myself. My name is Audrey Turley, and I've worked with Nelson Labs now for over 20 years. My area of expertise is biocompatibility, so in particular, all the in vitro tests. I also right now work in the technical consulting division of Nelson Laboratories, and I help to write biological evaluation plans for medical device companies, which is very appropriate when we talk about gas pathway devices, as there's unique considerations that need to be taken into account. And these evaluations can be taken to agencies ahead of time to get pre-approval to make sure that your plan of action will be fruitful in the end. And also, before we get too far in, I would like to just, take this moment to explain or maybe promote our new addition to Nelson Laboratories, which is a purchase of Toxicon Europe. And this is a chemical E&L laboratory, chemistry E&L laboratory based in Europe. And this is going to just expand our capabilities to do extractables and leachables. So we're really excited about that. If you have further questions, I'd like to direct you to our website at news.www.nelsonlabs.com. Okay, so the standards I'm going to talk about in my presentation today are, of course, ISO 18562, which is really a horizontal or a vertical standard when we talk about biocompatibility. So ISO 10993 is a broad horizontal standard that covers medical devices in general for biocompatibility evaluations. And 18562 is vertical, meaning it goes deeper into the risks associated with gas pathway devices. And then I would like to also reference a little bit of the US FDA guidance document on biocompatibility. There's not a whole lot of information in there for gas pathway, but it does drive us to use 18562. So when we talk about biocompatibility for any medical device, we want to start with a biological safety evaluation plan. This is our first initial step. So this is the big picture. I really like to step back and talk about what's applicable, how we're going to approach assessment of our devices. And so we start with this, biological evaluation plan, and this is really your initial risk assessment. This is what's asked for in 10993-1 for medical devices. So this is where we start. We move on then to carrying out the plan that we've laid out in our biological evaluation plan. So we do testing. We'll carry out risk assessments if needed, and address any manufacturing changes, because as we know, devices can go through several iterations between what you were told was your final concept and you decided to do biocompatibility testing, and then what actually gets released to market. So we can address those things in phase 2. And then the last phase is a biological evaluation report. This is asked for in 10993-1, specifically clause 7. Where instead of just submitting your reports and hoping that the FDA reviewer or your notified body reviewer can put everything together, you're going to do that for them and justify scientifically all your reasoning and your approach to your process. So when we talk about, our initial risk assessments, it's good to realize that that is where the conversation should begin. 18562 has a parallel title that is the same as ISO 10993-1. So it's evaluation and testing within a risk management process. So we can't drive this home enough that the agencies want you to start with what you already know and your specific risks of your device. So what you know about your device, what you know about your patient population, we're going to lay all of our cards out on the table. And the FDA has added an entire new section to their guidance document. So this guidance document sat in draft for like 3 years and everybody still followed it. But when they finalized it, they increased it by twofold. It was quite a bit more lengthy. And this was a valuable section in here to guide our thought process on how we're handling the risk assessments of our devices. The important parts I've underlined here that they want us to include are we are assessing material components. Now, this is not the FDA approving them, but it's what we are assessing as manufacturers and laboratories ahead of time. And manufacturing processes, and taking into consideration the clinical use of the device. And then from that, you take those and look at them from a biocompatibility perspective. I also like to put this definition up. This is from ISO 14971, how we're defining risk. So it is a combination of the probability of occurrence of harm and the severity of that harm. So how often can this harm occur? How often can this mishap occur? And if it does happen, how severe will it be? So it's good to start there when we're analyzing our risk. So I've put up this figure. This is from ISO 10993 Part 1. This isn't meant to be, read every word right now, but it's just to let you know that there is this flow chart that gives you a thought process when you're assessing your device. So we start with materials. We start with what we already know about the device. And we also can identify if we own this material or this process in another device that's already been approved. So it's good to leverage what we already have. This could reduce your amount of testing or let you know additional testing is needed, but it will benefit you for products down the road, possibly. In the past, we would typically go straight to this table. This is table A1. This is from the FDA guidance document. I like to use this as an example because it does put up FDA requirements and ISO requirements for medical devices. We would typically go here, categorize our device, and then move forward with our testing. So this checkbox approach really isn't appropriate when you're doing a risk-based analysis. We want scientific justification instead of saying, well, I did cytotoxicity because it's marked on the, on the chart. We want to say why it's necessary and what that tells us about the device. Another thing here is this is language to guide us to know that we need to use 18562. So FDA make sure to use gas pathway devices in their example of an indirect contacting device. So we're talking about tubes that are carrying air to a patient. I mean, they are not touching, for the most part, not touching the patient physically, but they are delivering things that go into the lungs. So FDA's use them in their definition of indirect patient contact. But ISO 18562 identifies that testing according to ISO 10993 Part 1 does not assess the appropriate risks for these devices. So we need to add some additional considerations. So for ISO 18562, this is just a summary of the standards that are incorporated under this. So there's 4 parts. The first one is still the initial risk assessment. So we do our risk evaluation here and assess the type of device, how it's used with the patient, our patient population. We're going to talk about our materials. This is all done in our initial step 1. Part 2, we're going to address particulates. That's a special risk to patients where it's going straight into your lungs on these gas pathway devices. Part 3 covers testing for VOCs, volatile organic compounds. And Part 4 covers tests for leachables in the condensate if you have a humidified gas pathway. So taking that into consideration, ISO 18562 has this flow chart here. I find it to not be 100% user-friendly, but it is some guidance for you to establish a flow for your process as you're evaluating your device and the steps you want to take. I spoke with the gentleman from the ISO technical committees that worked on this from ISO TC 194, and he has said a lot of these will be rewritten and revamped to make them a little bit more user-friendly. So I think we missed some industry input on these charts. But if you're a novice and you're looking for some guidance on a gas pathway, this is a good broad perspective and a place for you to start. So as we talk about the initial risk assessment, we offer these at Nelson Laboratories as a biological evaluation plan. These are the sections we find to be particularly important and that answer a lot of questions raised by agencies and notified bodies upon submission. And one new section, and it's not really new, but it's really been pushed down by the agencies to request, is a material characterization section. And this can vary depending on the contact of your device with the patient, but for breathing gas pathway devices, that's going to be pretty extensive because of the tests and the additional risks that need to be assessed. So we also include a device description and categorization. This is important because what you might categorize your device as would be maybe something different than the agency. And it's nice to have that laid out up front so that the agency isn't pointing back to that over and over again. They see where the disconnect has occurred up front in your submission. And then, of course, any special test sample preparations, which is going to occur with breathing gas pathway devices. So this is important. We can discuss how it was assessed, how the test setup will be done for the chemistry testing in particular. And then, of course, we would identify or lay out any testing and risk assessments that we intend to perform. So this is the initial document you should be having to cover you from the very beginning. So it should be this BEP on top of all your test reports to say this is why we did all of this that's following. So I like to come back out to this big picture view. So we've talked about our risk assessment, our biological evaluation plan, and then we move into the testing. So I'm just going to go over some of the general things for the different parts of 18562. And my input is based on our experts at our company, which we have a board certified toxicologist and our PhD chemists who have actually been in the ring a little bit with our clients in the FDA working this out. And there's a lot of disagreement in the industry about how these tests should be performed and what methods should be used, and working out the methods and the specifics with the chemistry testing laboratories. We have a long ways to go. And so that's the platform I'll be presenting from, just so you know that's based on our experience, because the guidance documents do not specifically state these. So it's really what we can come up with using the best science we have available. So this is the overview again for 18562. So the testing in particular, we'll talk about our particulates, VOCs, and then the measuring of condensates and what we recommend for those tests. So we start at the beginning by identifying the use of the gas pathway devices. If you have a dry air pathway, we recommend doing particulates and VOCs. So this is following part 2 and part 3. If you have a humidified air pathway, then we recommend following part 4, which is to measure your condensate. So there's a few things I've left on here. For dry air pathway, if you're going to do volatile organic compounds, compounds in air, you might have to do SVOCs, but it really depends on the agency. We have not seen that to be requested, but it is listed in 18562 part 3. And then again, for the condensate, we're looking for VOCs in water and metals in water as well. But we do have additional recommendations here that we've seen come back to be requested. And I'll go over those in specific. Before we talk about the specific tests, I always like to have this caveat up front that even if you start with a biocompatible raw material, it is important that we are analyzing this finished device, because processes and sterilization can impact this finished device. Clearly, it gets changed, things get added to it, the plastics change. So we want to make sure we're assessing this finished device. And it is stated in ISO 10993-1 to use a device in its final finished form, as final as possible. So if you are working things out with engineers, or you are an engineer, you can use this as leverage that you really need to have this final finished device for your testing. So particulates as part of 18562-2, these are required for dry air pathway devices. The standard lists 3 test options: a single particle filter, initial particle separators, particle counters. We have had FDA specifically request the NMAM 0500 with a 2.5 micron filter. That's just been our conversations with them before. It's not listed as a requirement in the guidelines, but this again has been the result of our interactions. There are limits set in the 18562, and they're based on US EPA 40 CFR Part 50. And the limits are based on size of particles. So clearly, the smaller the particulate, the further it's going to travel into the lung. And that causes a greater risk because it can be absorbed into the bloodstream, and then we have bigger issues than just lung interaction. And so we categorize the limits by size of the particles. And it's also important to recognize that this standard does not address nanoparticles that are coming off, nanoparticulates that could be coming off the gas pathway. So the limit is 0.2 in size here. It doesn't specify that in the guidelines, but I think it's important to note that. For the volatile organic compound testing, this specifically that we're talking about for the next couple of slides is for dry air. So the sampling is done differently. We do the gas pathway. In our experience, again, the FDA has requested this EPA canister method, which is TO15. It's then analyzed by gas chromatography and mass spectroscopy. There are other test methods available, but sometimes the data is hard for the toxicologist to interpret and then apply to the patient population. So we've seen TO15 be the most requested, and that has worked for the FDA. They've accepted that. In the guidance document, it does reference to use ASTM D5466, and then it states these acceptance criteria, which are based on the threshold of toxicological concern. So I just like to take this time to point out that you really need a toxicologist, a board-certified toxicologist, to assess and interpret these results, and so that you're applying these thresholds appropriately. And we'll go through some of the calculations so that can be made a little more clear further on. But these acceptance criteria are based on breathing rates that default breathing rates are published in Part 1 of the 18562 standard that they're basing these off of. For the condensate, this is in Part 4, so these are required for all humidified air gas pathway devices. So this section is really anticipated to be rewritten, and our conversations we've had with the gentleman who heavily wrote this and collaborated the effort of the writing, that some of the recommendations don't fall in line with what's appropriate. One thing to take in consideration is that you can assess the leachates of this device by using ISO 10993 testing. So if you've already done that testing on your device, you don't need to go through and do this condensate testing. However, if you haven't done that, you can use some of this testing in lieu of ISO 10993, and this does tend to translate better to clinical use of the device. So one thing we like to really promote here is to make sure you put this testing plan together, take it to the agency in a pre-sub, because there is so much disagreement in the agencies right now. You should get some forethought going before you start doing all of your testing. Save yourself some time and money in the long run. So I just want to talk a little bit about how extractables and leachables work. One thing I forgot to mention is when we assess the condensate, our typical recommendation is to do extractable and leachable. So we call it a full panel, and I'll explain what that means. But instead of doing specific testing on a condensate that accumulates in the product, which is unrealistic, which is actually what's mentioned in Part 4, we recommend doing an extractable and leachables. So typically what we do is we run the extraction, we'll immerse the device in water for 50 degrees for 72 hours. We do just recommend using water because that is what's clinically relevant at this point. We have had success in promoting that to the FDA. So I think I'm going to say this over and over and over again, that it always depends on your reviewer in the FDA. That's why a pre-sub meeting is great. Submit your biological evaluation plan. Point these things out, because the FDA in the past or with other extractable and leachable testing, they'll want you to use a polar, a non-polar, and sometimes a semi-polar solvent. So it's important to have this conversation up front. So what are we looking for when we run these extractable leachables testing? We really cast a wide net, analyzing for anything we can find in the condensate or this would be in the extraction. So we look for inorganics, which would be metals, and then organic solvents, which would be our volatiles, our semi-volatiles, and our non-volatiles. So for the organic compounds, it's really size-based, this separation. So if you have volatiles and semi-volatiles, we'll use a GC, gas chromatograph, gas chromatography, because those are smaller and they will evaporate. And then for non-volatile organics, we do use an LC, a liquid chromatography. This is an example of a gas chromatograph, but it's the same idea for the liquid chromatography as well. So what we see here are compounds that get sorted out by size, really. So it's the length, the x-axis is how long they stay in the machine, and then the y-axis is an abundance, so how much of it's in the machine. So that tells us, breaks it down by size, and then we can identify it further by using mass spectroscopy. So here on your left, you see the results you get for chemistry. These really don't translate to a patient, so you can't stop here. You have to go further and have a toxicological risk assessment. So you can assess maybe you have a special population that this is used for neonates, and maybe you want to categorize it for all the populations, adults, pediatrics, neonates, so you need to translate that to all the different populations. So you take those results and then you analyze them through a toxicological risk assessment. And I've just broken this out into 3 easy steps. It's really not easy, but these are 3 general steps, I should say. If you're a toxicologist, don't throw anything at me. I know it's not easy. So the first part is you determine your chemistry and you translate those results into micrograms or milligrams of device. This is what unit you use depends on what you're going to use units for your research data and for your calculations. So you translate that to amount per device, and then you're going to research available toxicological data that's already published. This is where you save some time in animal testing as well. If you do this, you can already use animal testing that's been performed and published for the specific compounds that you found on your device. So this really relates well to your actual device. And we're looking for NOELs or LOELs. We'll look for reference doses as well. But NOELs and LOELs are these levels that are established in the experimental tests where there's no adverse effect that was seen on the subject animal. We use those values and then we run calculations according to ISO 10993-17. So you can see where 10993 and 18562 really do work back and forth to run these assessments. So you cannot use 18562 without using ISO 10993 as well. So we'll calculate a tolerable intake, a tolerable exposure, and in the end, we're going to calculate a margin of safety. And this bottom line, that's where everything comes down to, is this last calculation. And I've just summarized them quickly on this, really short on this page, on this slide. For our tolerable intake, we take the NOEL that we found from our published literature. This is the first step of our calculations. And then we take into account some utilization factors. That's what this UF1, 2, and 3. So we have to take into account variable things. How well does animal data translate to human use, right? How good is our research? How good is the data that we're finding published relate to our use of the device? So most of the published literature is not going to have a lot of inhalation data for some of these compounds. So we might find something that had like an oral exposure. Well, that doesn't translate very well to an inhalation exposure. So we adjust our utilization factors. The toxicologists adjust them appropriately, understanding the appropriate risks. And then, of course, there's inter-individual variation that we will see. And there are default values that are set for a couple of these that are published in 10993-17. And then we get a value that equates to so much of this compound per day that a patient or a person could be exposed to, where we wouldn't expect to see anything adverse. Then we use that number and we plug it into our tolerable exposure. This takes into account our specific population, and that's done by the inhalation rate here that's measured. So if we have, pediatric patients, their breathing rate and breathing volume is very different from an adult. So we want to take that into account. And then we have this utilization factor. That's another correction factor that's done to account for different areas we could be exposed to the same compound. If you find something very common like acetone, we're exposed to that in other places, especially in a hospital setting, right? And then we also add in another part of that calculation for the UTF is how many different devices could be used on the patient that have the same compound in it. So it sounds like guesswork, but there's education that goes behind the decisions that are made for these adjustments. So this is just an additional point to bring out that a board certified toxicologist is needed to run these calculations and to perform the correct adjustments in these factors. So then that brings us to our margin of safety calculation. This is where we're going to now apply what we found in published literature. We've related it to your patient population. Now we're going to bring in the medical device. So we have our tolerable exposure, and then we're going to divide that by the amount that's actually coming off the device and obtain a ratio. So if you have a margin of safety that is greater than 1, that indicates a low toxicological hazard. I will just put a little warning out here. If you have a special population like neonates and you have a margin of safety of 1 or 1.1, that may not be an appropriate level of risk still for that patient population. And it depends on how conservative your toxicologist is, but you may want to go back and see if something can be adjusted, if you can do some targeted chemistry testing to further assess the compound that's still borderline raising some red flags. So in the conclusion for the toxicological risk assessment, we'll make sure to point out that the risk assessment is based on chemical characterization data, literature research, and your derived margins of safety. So again, we come down to that last calculation, that margin of safety ratio. And the hope is, this is a dream toxicological conclusion, that this risk assessment indicates that the likelihood of adverse effects from the device is considered low for all compounds. So this assessment not only occurs if you do a full extractable and leachables for a condensate or a sample or a product that has humidified air, but it also applies when you do VOCs for a dry air pathway. You're still doing a toxicological risk assessment, running these calculations, driving to this last conclusion here. There are some additional considerations. And I said it before that you might have possible follow-up analytical testing based on the compounds that were identified, the levels, your patient population, and sometimes because of the test procedure that was selected, there might be some things that need to be tweaked and adjusted. This is still an area of very finite science where everything is down to the last detail. And so there may need to be some additional considerations once we see what comes off on the results. Another thing to take into consideration, if you have components that also touch the patient's skin, you have a face mask, you do need to do biocompatibility testing according to 10993 Part 1 that's outlined there. Typically that includes cytotoxicity, irritation, and sensitization testing. So I'm just going to go over those tests very briefly as well. For cytotoxicity, we're looking for, any toxic effect on cells. This is a really nice view of some cells, good rounding, good attachment. This would be a 0, which would be passing for cytotoxicity. And here we see a complete severe cytotoxic reaction where we see significant cell death. So for cytotoxicity, on the scale of 1 to 4, this is a subjective test. So this is based on an analyst's interpretation of what they look through the microscope and see. So they're just basing this score off of their interpretation of the view that they can see of the cells. And if we have a 2 or lower, that's considered passing. If you're above a 3, then you're failing the cytotoxicity test. So then we would run an irritation test. This is typically done as an intracutaneous reactivity test. This is injection-based test. So again, we're extracting medical devices for these tests, and we're exposing the extract to these test systems. So for here, we inject a small amount of fluid under the skin of a rabbit, and we analyze that at 24, 48, and 72 hours. The goal is when you're compared to the negative device or negative control that you are 1 or less as far as a comparison in reactivity. And for sensitization, I like to put this image up because it's from a textbook. It just shows that this is a very complex response. And this takes about 60 days to run this test because you have to have a couple different exposures to the animals to have a full systemic response. And there's a few test options here. We do a guinea pig maximization, a local lymph node assay, or a Buehler method. For the guinea pig maximization and the local lymph node assay, these are the most commonly selected tests that can be performed. But it depends which test. You can see there's a savings in time. If you could go from 6 weeks, if you go from 8 weeks of testing to 6 weeks, you would probably choose the local lymph node assay. The problem with that is local lymph node assay is only accepted in Europe for a CE mark. The US FDA does not accept this typically. They like to see the guinea pig maximization. So if you find yourself doing the sensitization test and you're going to submit to both markets, do the guinea pig because it is accepted in Europe as well. So we step back out to this big picture. We've done step 1, we've done our testing, we've done our risk assessments, our chemistry testing, and now we're going to do a biological evaluation report. This sometimes gets a little bit of oversight, I think, and they just have, the engineer who was in charge of coordinating all the testing is just going to write everything up. But what we're finding is that agencies are coming back and saying we need somebody with the proper education and experience to say your device is safe. So they want somebody with some biocompatibility background, a toxicologist if necessary. Maybe you didn't do a separate toxicological risk assessment, but you need to have that input. You need to have that expertise reviewing these reports, or you will get kickback from, we've had it the most from DECRA, honestly, that they are looking for this. And it's a great place to talk about all the setup, justify what was done in the chemistry testing, how you analyzed your sample, how which method was chosen. That's really important, especially now when there's so much disagreement in the agencies where we're not having consistent feedback about what's expected. So we also do biological evaluation reports. And I like to put this table of contents because it does make you aware of all the things that can be included. And this is where you want to have these conversations with the agency. So again, we would do like a material characterization, talk about your materials. Again, you cannot state this enough in your presentations to them. Talk about your biocompatibility tests. I put this particulate discussion in here because sometimes we see particulates in biocompatibility extractions. Have a conversation about that. Talk about, maybe do some additional particulate testing on those extracts and eliminate or mitigate the risk right here instead of waiting to see if they catch it. Just have the conversation up front. Talk about your chemistry testing, your toxicological risk assessment, and then, of course, a material change assessment because it was changed once again, right? Between the testing completion and submission, you can have this all wrapped up in one package for the agency. And your conclusion at the end should be that the device is biocompatible and meets the requirements of 10.9.3 Part 1. And we would add for gas pathway that it also meets the intent of 18.562, assessing all the risks of the device to the patient. So now I can open the floor for questions if you guys have any. I'd also like to say if you have some very technical questions, especially about the chemistry testing, that is not my area of expertise about the details on the chemistry testing, but we can offer you a 30-minute free consultation with our experts, and we can set that up back at our booth with our sales rep, Jackie. And we're at 2128 if you want to stop by and get some more information about that. Any questions? Okay, great. Well, feel free to stop by our booth at Nelson Labs and we'd be happy to discuss your biocompatibility. Thank you.